Well, thank you for that introduction, Steve, and thank you everyone for coming. I'm really excited to be sharing with you what I've been working on for the last several years. My talk is entitled The Landscape Epidemiology of Zoonotic Diseases, Rodent-borne Bartonella and Livestock Anthrax. Over the course of my time here at Stanford, I've become increasingly interested in landscape epidemiology, or mapping and modeling the conditions that govern pathogen spread, and in zoonotic diseases, or diseases that people acquire from animals. I've become increasingly interested in how these relate to the growing threat of emerging infectious diseases. We're living in a time of increasing emerging infectious disease events, or identification of new or resurging infectious diseases. Some of these you might recognize, like Lyme disease, West Nile virus, dengue, and yellow fever. This is a chart showing new or emerging infectious disease events by decade. And as you can see, zoonotic diseases represent a growing proportion of increasing emerging infectious diseases. Zoonotic diseases represent a growing threat to public health. The epidemiology triad is one way to look at the spread of zoonotic disease. Humans become infected when they come with pathogens, when they come into contact with reservoirs such as livestock, and a pathogen could be Bacillus anthracis, the causative agent of anthrax. The emergence of zoonotic diseases is often attributed to anthropogenic changes, such as changing land use. And some argue that an increase in land use change might actually be driving emerging infectious disease events. Indeed, we are also living in a time of unprecedented landscape change. Here are the stats. From 1980 to 2000, over 80% of a 100 million hectare increase in agricultural land came from forests. And between 2000 and 2012, total global forest cover decreased by 16%. We know that changing land use must somehow impact zoonotic diseases, but we actually know very little about the impact of land use in several disease systems. This is what I'm trying to figure out, how the environment impacts zoonotic diseases. But what we do know is that these diseases are maintained in complex cycles that comprise pathogens, reservoirs, and humans, and this all occurs in an environmental context. My work focuses on understanding this environmental context of disease and what it means for human health. Now, when an environment is changed, people come into contact with reservoirs and pathogens they may have previously had no contact with. This interaction, combined with changes to the landscape, alters the natural focus of disease, which is where these three things overlap. At the natural focus of disease is where infectious diseases emerge. There are two major categories of zoonotic diseases, diseases from wildlife and non-wildlife. In order to understand these two types, let's look at the areas of greatest risk. This is a map depicting emerging zoonotic disease events from wildlife over 1940 to 2004, and the red areas represent an increasing risk of emerging infectious disease. East Africa, and Kenya in particular, is at increased risk for an emerging infectious disease event from wildlife. This is why one of my two case studies focuses on Kenya. This is a similar map, but depicting emerging zoonotic disease events from non-wildlife. And as you can see, Bangladesh is highlighted in bright red. My dissertation work comprises two case studies of zoonotic disease emergence. A rodent-borne pathogen in Kenya, bartonella, where the resulting disease is from wildlife. And in Bangladesh, we have a livestock disease, anthrax, a disease from domestic animals. Both diseases are caused by bacterial pathogens, and both are recently emerging zoonotic diseases for their localities. In addition to looking at this from an epidemiology lens, my approach is interdisciplinary, so I also include a disease ecology lens. Disease ecology includes the population interactions that govern the spread of disease. Now, in researching the landscape epidemiology of zoonotic diseases, I found there are essentially two pathways by which to understand how the environment impacts zoonotic diseases. And that is from the top and bottom of the following conceptual diagram. Here's how I understand the impact of the environment on zoonotic diseases. Classic epidemiology takes human cases of disease and identifies human behavior associated with those cases. I'm calling those proximate determinants of disease. There are certain animal cases associated with human cases, And upstream of that, there's an underlying species distribution from which these animal cases arise. And if we take a step further, there are underlying determinants of a species distribution, and I'm calling those distal determinants of disease. You could also imagine working in the opposite direction, starting with distal determinants of disease and working towards human cases to understand how the environment impacts zoonotic diseases. So that leads me to my first question. How does land use impact rodent abundance and diversity? And my second question, how does land use impact rodent infection with bartonella? And my third question starts with human cases of disease and works in the opposite direction and asks, what are the environmental attributes associated with livestock anthrax in Bangladesh? So as I mentioned earlier, Kenya is an area at increased risk for an emerging zoonotic disease event from wildlife. Febrile illnesses are commonly reported in people living in rural areas in Laikipia County, Kenya, which is pictured here on the left in green. Zoonotic diseases often present clinically as prolonged or relapsing fevers or febrile illnesses, but their cause remains largely unknown in this area. Rodent-borne diseases could be a potential source of febrile illness in Laikipia. Laikipia is a tropical wooded savanna. It's considered one of East Africa's most important areas for wildlife conservation. It is also an area under pressure from mechanized agriculture, intensification of rangeland use, and expanding subsistence agriculture. It is also an area that contains a patchwork of land use types, also making it an ideal location to examine how land use might influence the risk of rodent-borne diseases. But before we delve into research questions, I want to talk about a couple of concepts that will help us frame the results in the context of the literature. There is one major concept that informed this segment of my work, and that's the concept that increased abundance leads to increased disease transmission. So holding prevalence constant, which is the proportion of individuals infected, holding this constant, an increase in abundance should lead to an increase in disease transmission. Thus, if we know what governs rodent abundance, we can use this information to predict when and where we might see large increases in rodents, and subsequently what areas or times are of greatest risk to humans. So what governs rodent abundance? There are several factors that govern rodent abundance, and they depend upon location. Rainfall and plant cover have been correlated with rodent abundance in the American Southwest. And this is through the trophic cascade hypothesis, which states that increased rain leads to increased primary production, which leads to a boom in the rodent population, and then an increase in the density of infected rodents. Soil textures have been correlated with rodent abundance in Central Asia, in particular clay soils. And rainfall and large mammal defaunation have been correlated with rodent abundance in Kenya. Now, defaunation is the loss of large wildlife, and it leads to rodentation or a boom in the rodent population. In experimental exposure studies in Kenya, a decrease in large wildlife has led to an increase in the rodent abundance and then a subsequent increase in the density of infected rodents. So it's unknown how large wildlife regulate rodent populations exactly, but at least for grazing ungulates, regulation is likely to be through competition over food resources or other mechanisms. This shows that there are 4 factors to look at: rainfall, plant cover, soils, and large mammal defaunation. Now, defaunation in the real world often occurs with land use change. So my first research question asks, how does land use impact rodent abundance in Laikipia, Kenya? This is what led me to hypothesize that agricultural areas would have the highest rodent abundance and present the greatest risk to humans, partly because of the greater availability of food, and also there would be less wildlife due to defaunation, so they would face less competition. And just to remind you, this is where the first research question comes in: what determines species distribution? So to answer this question, I used rodent trapping data from my collaborator, Hilary Young, to build a negative binomial model. These data are divided into what I'm calling the early season and the late season. The early season goes from January 2011 to May 2011, and the late season from June 2011 to October 2011. They're divided into these 2 periods based upon the bimodal rainfall pattern in the region and based upon work from others in Tanzania that had success with predicting rodent number following each rainy season. I then used satellite data to characterize vegetation from the area, and I used the Normalized Difference Vegetation Index, or NDVI, which is just a transformation of the spectral imagery to highlight green vegetation. And this is a map of the NDVI in the early season and the late season with rodent trapping sites, so you can see some of the variation. I then summed NDVI from October to December for the early season and summed NDVI from October to June for the late season. I also included soil texture data as represented by sand to silt ratio as an indicator of underlying vegetation productivity. I also included mean annual precipitation, which I interpolated from 73 rain gauges in the area using a Gaussian co-Kriging model and using elevation as the covariate. I then used a map from Impala Research Center, which includes ranching and wildlife areas, pastoralist grazing areas, and mixed farming areas. The sample sizes for each model are 42 for the early season and 37 for the late season. And I picked the best fit model for each by using the lowest AIC, which is just a measure of the relative quality of the model. And I chose these 4 factors: rainfall, vegetation cover, soils, and land use, based upon those 4 factors that have been identified in the literature as being important. So of the 4 potential factors, the best fit model was the one with sand to silt ratio and land use. Here's what we found. So you have sand to silt ratio on the x-axis and number of rodents on the y-axis, and you have ranching and wildlife in red, pasture in blue, and farming in green. And what you can see is for increasing sand to silt ratio, rodent abundance increases in all land use types, but this relationship is much steeper for ranching and wildlife. Now, these results are not completely straightforward, so I'm going to give you a couple of interpretations. First, this relationship of increased silt ratio and rodent abundance corroborates findings from another group in Tanzania, which found that rodent abundance was higher in coarser soil textures. And the thought being that soils with lower sand and higher clay content become waterlogged and less amenable for burrowing. Second, the difference in slope for farming and pasture may be due to human modifications of the environment, such as via land cover or predation by domestic animals such as cats or dogs. And that might weaken that relationship that we saw between soil texture and abundance. In other words, the rodent population could be regulated by land cover or predators. So we thought that areas of greatest risk would be agricultural areas, but were in fact wildlife and ranching and pasture areas at coarser soil textures. We would need more data to fully understand these relationships among sand to silt ratio, land use, and rodent abundance, but these data imply that wildlife and ranching and pasture areas should be under more observation for rodent-associated diseases. Out of these 2 populations, only one likely has consistent access to the health system, those living in ranching and wildlife areas, because those employed through ranching and conservation enterprises tend to have employer-sponsored healthcare coverage as compared to pastoralists and farmers. And if we project these model results onto the landscape, this would be our map of risk, with green showing increased risk for greater rodent abundance. So in conclusion, rodent abundance is highest in areas with coarser soil textures, but this was mediated by land use. This relationship between increasingly coarse soil textures and rodent abundance may be the result of coarser soil textures being more amenable for burrowing. And the dampening of this effect by land use type may be the result of differences in land cover and use or predation by domestic animals. And ranching and wildlife areas and pasture areas at higher sand to silt ratio are of greatest risk, and thus warrant further study into rodent-associated diseases in humans. Now that we know what governs rodent abundance in Laikipia, Kenya, what governs abundance of infected rodents? That leads me to my second question. How does land use impact rodent infection with bartonella, a rodent-borne pathogen? Rodent-associated bartonella have been found in rodents and their ectoparasites in Laikipia County, Kenya. What are bartonella? They're bacteria that can cause febrile illness and other clinical outcomes in humans. Rodents are the primary reservoir for at least 2 known human pathogens from this genus, and fleas are believed to be the primary vector of rodent-borne bartonella species. Now, there are two ways to measure rodent infection: prevalence and density of infected rodents. Prevalence is just number with Bartonella over number tested, and abundance of infected rodents or density of infected rodents, I'm using those interchangeably, is prevalence times the number of rodents per hectare. And today I'm going to focus on abundance of infected rodents because it more closely approximates human risk. So as we discussed earlier, defaunation, or the loss of large wildlife, leads to rodentation. So a decrease in large wildlife leads to a near doubling in the rodent population, which leads to an increase in the density of infected rodents. Also, the presence of livestock reduces rodent abundance by the same amount as large wildlife. So an increase in cattle leads to a decrease in rodent abundance. So that leads me to my hypothesis. The abundance of infected rodents will be regulated by herbivorous competitors in wildlife areas and in pastoral areas. So by wildlife in wildlife areas and by livestock in pastoral areas. To investigate this, I used the same model in covariates as the previous rodent abundance model. And among those 4 factors associated with rodent abundance in the literature, only mean annual precipitation, or in other words, rainfall, was associated with abundance of infected rodents in the early season. And in the late season, mean annual precipitation by land use, the interaction of those 2 variables, was significantly associated with abundance of infected rodents. So this is a graphical representation of the model results. You have mean annual precipitation on the x-axis and abundance of infected rodents on the y-axis, and this is the original data with the fitted regression curves on top. And you have wildlife and ranching in red, pasture in blue, and green in farming, or farming in green. And what you can see here is an increase in rain. An increase in rain leads to an increase in rodents. And this relationship, increased rain, increased rodents, corroborates findings from the trophic cascade hypothesis, which states that increased rain leads to increased primary production, which leads to a boom in the rodent population. So this relationship is consistent with the trophic cascade hypothesis. However, pasture does not follow this relationship. But as you recall from the earlier study, what mattered for overall rodent abundance was soil and land use and not rainfall. So I looked at a second model with a close AIC to see if soil and land use were also important for infected rodent abundance. And I found that sand to silt ratio by land use was significantly associated with abundance of infected rodents. So what you see here is sand to silt ratio on the x-axis and abundance of infected rodents on the y-axis. And we see that with an increase in sand to silt ratio, abundance of infected rodents increases for all land use types, but this relationship is much steeper for wildlife and ranching. And this looks largely similar to the results for overall infected rodent, or overall rodent abundance, which is shown here. So combining the results of these 2 models, the major difference between rodent abundance and infected rodent abundance is this effect of rainfall. Why does rainfall appear to be an important factor in infected rodent abundance, and it wasn't in overall rodent abundance? Well, while soil texture and land use are able to explain rodent abundance, precipitation may be more important in determining the prevalence. Indeed, survival of all pre-adult stages of fleas are thought to be governed by high relative soil humidity, which increases with precipitation in arid areas. So mean annual precipitation may have more to do with flea survival than rodent abundance. And if we project these model results onto the landscape, here is our risk map for density of rodents infected with Bartonella. So in conclusion, mean annual precipitation was positively associated with abundance of infected rodents in the early and the late season, though this relationship was mediated by land use in the late season. That main effect of precipitation seems consistent with the trophic cascade hypothesis. However, I argue that this may in fact reflect factors that govern flea survival. Now, what does this mean for human health? Well, areas of greatest risk are high precipitation wildlife and ranching areas and low precipitation pasture areas. And as healthcare access is likely greater for people employed in wildlife and ranching enterprises, we may actually be capturing one of the populations most at risk. However, increased surveillance for Bartonella species may be warranted in low precipitation pasture areas. So as we discussed, people can become exposed to pathogens via wildlife, but they can also become exposed to pathogens via the animals that they tend. To look more closely at this, we're going to Bangladesh, which is at increased risk for an emerging zoonotic disease event from non-wildlife, in this case, livestock. My first two projects dealt with the distal determinants of disease, and my third question starts with human cases of disease and asks, what are the environmental attributes associated with livestock anthrax in Bangladesh? So in August of 2009, a researcher at the International Center for Diarrheal Disease Research, Bangladesh, was contacted by his relatives living in Pabna district, and this was concerning several people in his village who had contracted skin lesions following the slaughter of 2 sick cows. This call prompted an investigation into a potential outbreak of cutaneous anthrax, or anthrax that's characterized by skin lesions. Over the course of the next 2 years, there would be 14 outbreaks in total. And following these cases, a multidisciplinary team investigated the outbreaks, and they found that those who had participated in slaughtering a sick cow were at 22 times greater risk of developing cutaneous anthrax as compared to people who didn't. So from these investigations, it became clear how people were getting anthrax, but it remained unknown how cattle were getting it. Bacillus anthracis is the causative agent of anthrax. It's a spore-forming bacteria. Transmission to livestock is believed to occur through ingestion of Bacillus anthracis containing soil or water. And the vegetative form of Bacillus anthracis that undergoes replication in the environment is believed to be shed at death or during slaughter into the environment. And the vegetative form sporulates on contact with oxygen. So you might be wondering, how do we typically deal with livestock anthrax? Well, there's a vaccine, and 2 laboratories in Bangladesh produce this vaccine. However, the total ruminant population in Bangladesh is 48.7 million. So for a low-income country, spending limited government funds, not to mention logistical issues, may make cattle vaccination difficult. Therefore, it's important for us to understand the context in Bangladesh, such that we can either target vaccines more effectively or develop new options for disease control. Right now, there are 3 hypotheses in the literature for what causes livestock anthrax outbreaks. So we know from the literature that Bacillus anthracis occurs naturally in the soil in other places. It's predominantly a hot season disease of hot, dry climates. And outbreaks are also correlated with alkaline soils and soils high in organic matter. And the hypothesis in those systems is that hot, dry weather stresses livestock and allows low doses of spores to become infective. And this is the first hypothesis. Hot, dry weather stresses livestock, making them more susceptible. However, though in a minority worldwide, there are also outbreaks that occur during wet seasons. And the hypothesis in those systems is that during wet season, soil moisture is high enough so zebra pull out entire clumps of grass while they graze, so they eat more soil. And this is from Etosha National Park. So on the one hand, we have stress that makes cows more susceptible, and on the other hand, we have rainfall and high soil moisture that makes soil exposure higher. And this is the second hypothesis. During wet season, soil moisture is high enough to facilitate pulling entire plants from the ground. Now, despite consensus on anthrax being a soil-borne livestock disease, the prevailing wisdom in Bangladesh is that these outbreaks are caused by contaminated feed. And the idea is that recycled anthrax carcasses are put into concentrated feed. Now, the authors of this study believe that the environment in Bangladesh is not permissive for Bacillus anthracis. And this is based upon 2 pieces of evidence. One, the high genetic diversity of strains found in those outbreaks, and it was the monsoon, so cows couldn't have been grazing. Given these 3 hypotheses, what is the source of livestock anthrax in Bangladesh? This brings me to my first trip to Bangladesh. This is an image I took in Shirazganj, the first time I accompanied the outbreak team to the field. And this is when I began working with the team to understand the circumstance of livestock anthrax. Well, first we can already throw out the bone meal hypothesis because a few months after that bone meal study, my colleague Saiful Islam in Bangladesh found that there were no rendering plants for bone meal and that livestock were fed locally produced feed. So they couldn't have been eating contaminated feed from outside of Bangladesh. And contrary to the feed hypothesis, recent evidence from a study led by my colleague Najmul Haider, we found that it was not concentrated feed that cows were exposed to, but soil. we found that cows were actually grazing, and the longer they spent grazing, the more likely they were to develop anthrax the closer they grazed to the ground. And we also found that people were bringing grass from the fields to cows that were kept in sheds. And if people brought this grass, which was different from concentrated feed, we found that livestock were more likely to get anthrax. Interestingly, if people washed this grass, presumably washing off the soil, they were less likely to have an anthrax case. We also found that people were collecting soil from agricultural lands and adding this soil to the bottom of their cow shed. And if they did this, they were more likely to have a farm with an anthrax case. So contrary to the feed hypothesis, this evidence suggests that soil exposure is the source of livestock anthrax in Bangladesh. So hypotheses 1 and 2 are likely at play, but we still don't know if it's the wet season or dry season transmission. Do the outbreaks have more to do with soil properties, such as in dry season transmission, or timing of rainfall, such as in the wet season transmission? To answer this question, I used livestock case and control data from my colleague Najmal Haider. Now, anthrax can cause cutaneous, gastrointestinal, and inhalational anthrax in humans, and cutaneous and gastrointestinal are the forms that occur in Bangladesh. Suspected or probable human cutaneous or gastrointestinal are reported to the Institute for Epidemiology, Disease Control, and Research, and they're reported through 3 channels: government health channels, hotline, and media-based surveillance. And livestock cases are identified in connection with human cases. So they investigate the human case and then look for the antecedent livestock case that caused the human case. So I did a case control study from 2012 to 2016. I defined an anthrax case, and this is a picture of a likely anthrax case. So a case is any domestic ruminant having sudden death within 24 hours of onset of clinical illness. And controls are geographic farm controls that are disease-free and enrolled as the 10 farms north, south, east, and west from the case farm. And GPS coordinates were taken of case and control farms. And I defined the study area as subdistricts with the presence of suspected or a probable livestock anthrax case over 2012 to 2016, and as those districts that lie within 30 kilometers of those districts. I digitized soil maps from the Soil Resource Development Institute in Bangladesh to characterize soil attributes that might be correlated with outbreaks. These are soil texture, pH, and organic matter content. I then used data from the Climate Hazards Infrared Precipitation with Stations data over 2012 to 2016, and that's gridded rainfall data. I then used soil moisture from the Soil Moisture Active Passive Satellite to characterize soil moisture in the region. And why did I digitize soil maps? Remember from the literature background, alkaline soils and soils high in organic matter were correlated with outbreaks globally. So I looked at those 2 attributes in these outbreaks. And what did we find? With respect to soil attributes, we found no clear relationship of livestock cases and organic matter. So you have organic matter content in percent on the y-axis and case and control on the x-axis. And as you can see, there's no significant difference between the 2. Further, we found no clear relationship of livestock cases and soil pH. You can look at this as 2 overlapping histograms. This is pH on the x-axis and count on the y. Controls are in red and cases are in blue. And there's no clear relationship as cases occur at all pH values. But what we did find is that there are outbreaks that occur on soils of a pH less than 6.1, which hasn't been identified outside of Kruger National Park, South Africa. So this is a novel finding. I then wanted to dig deeper into the timing of cases. And I observed what visually appears to be seasonality of livestock anthrax cases. So this is an epidemic curve. You have years on the x-axis, cases per month on the y. So these blue boxes are the cases, and the line is rainfall in millimeters. And what you see here is an increase in cases with the monsoon and a decrease in cases with the end of the monsoon. And that's a pretty clear relationship for most years. Now, anthrax is typically thought of as a dry season disease of dry climates and has only been found to coincide with the wet season in Etosha National Park, Namibia, in outbreaks in zebra. So this is also a novel finding. So I wanted to dig a bit deeper into this relationship. This appears to be a strong visual trend, but does this hold up quantitatively? So I calculated and graphed the cross-correlation function of monthly precipitation and monthly livestock anthrax cases. So you have lag in months on the x-axis and autocorrelation function on the y. And the way you read this graph is it varies the x value, which is rainfall, and holds the y value, which is livestock cases, constant. So a lag of 0 means that livestock cases at time 0 and rainfall at time 0 have a correlation of about 0.4. And a lag of +1 means that livestock cases at time yt and rainfall at time t+1 have a correlation of about 0.5. So the blue lines are confidence intervals, and what we see is a statistically significant sinusoidal pattern, which indicates two things. One, periodicity to the data, and two, that these two variables are correlated. So we have no relationship with measurable soil attributes, but what we do see is transmission during the wet season. It's unclear what the mechanism is. One possible mechanism is that increased soil moisture allows cows to pull up and ingest entire clumps of grass and soil while they're grazing. And as you can see here, you have another epidemic curve. This is cases per month on the left-hand y-axis, and soil moisture is the line. And what you can see here is an increase in cases with increased soil moisture and decrease in cases with decreased soil moisture for 2015. But for 2016, we don't see much of a relationship, especially for April. So there's some indication of a relationship, but there might be something else going on for 2016. And so our results appear consistent with the wet season transmission hypothesis that the growing season is the time of increased risk, but we really can't speak to mechanism. So this information is useful for two reasons. One, it signifies an environmental exposure pathway for anthrax transmission. And secondly, this information is useful because Bangladesh has a strong government public health organization and strong public health research organizations with ties to livestock organizations. And they really have the capacity to respond to outbreaks. And so what this evidence really points to is a seasonal administration of livestock anthrax vaccine such that protection is conferred during the monsoon season. So in other words, this research suggests that livestock and government organizations could vaccinate cattle before the monsoon. So I hope I've left you all with an appreciation of how our environment might impact our risks of emerging zoonotic diseases. In summary, you saw two case studies of zoonotic diseases, a rodent-borne pathogen in Kenya and a livestock disease in Bangladesh. In both, we saw the importance of the environment on determining the timing of cases and the distribution of reservoirs. And what this work highlights is the ability of the interdisciplinary perspective to help us understand the broader distal environmental determinants of disease and what this means for human health. And as shown in these two cases, we see how understanding these determinants informs disease control strategies and improves our ability to combat zoonotic diseases. And with that, I'll go through a couple quick acknowledgments, and then I'd love to take your questions. So first, let it be known that I have the best committee. Jamie Jones, Michele Barry, Lynne Gaffikin, Steve Luby, Eric Lambin. Thank you so much for your support over the last several years, for your critique that's made me a stronger scientist, and for being just excellent mentors. I've learned so much from you all. I'd also like to thank Nicole, who's our chair. Thank you, Nicole. I'd like to thank the E-IPER program and the E-IPER community. Thank you all. It's really a one-of-a-kind program, and I'm really proud to be a part of it. Also like to thank my funders, David and Lucille Packard Foundation, E-IPER Summer Funding, McGee Leverson Grants, and also Eric Lambin and Jamie Jones. Like many E-IPER students, I had several labs to call home. So thank you to the Jones Lab, the Luby Lab, and the Land Change Lab. I see so much for your critique and for your friendship. A lot of people have been really generous with their time. Scott Fendorf, Evan Lyons, Rodolfo Durzo, the Hume Center and Graduate Life Office for their support, my friends in the ESS community, I'd like to thank the ICDDRB, in particular the anthrax outbreak team, for showing me how to do outbreak investigations. I have a picture here of the team working. I think they're swabbing bone marrow for anthrax. And thank you, Bilal, who's Shamshur Rahman, who was my field assistant, and Farhana Haq, who was our team leader. And this is my primary collaborator. And I'd like to thank Hillary Young at UCSB for being a great collaborator and mentor. And many, many friends. I'm not going to list all of you, but thank you so much for keeping me grounded and for all of the fun over the last several years. You really made my time here at Stanford. And a couple of you are up here. Your beautiful faces. I feel really fortunate to be surrounded by such wonderful people, so thank you. And a couple people, Ashley Hazel, Honoré Pedigo, and Netta Orezi. They've been my urban family for the last several years, and they've been there through everything, and you guys are truly wonderful, so thank you. And I'd like to thank my family, my mom and Mike, who are here, and my dad, who's on Skype, who listened to this presentation I don't know how many times over Skype. So thank you, Dad. And also like to thank Lillian, Blake, Virginia, Mark, and Terry, and Dave for their support. And last, I'd like to thank my partner, Danny, who couldn't be here, but is very excited that I'm finishing. So thank you, Danny. And your questions.
So, great presentation. It's really interesting stuff. For your last question, you used soil mapping data, right? Do you have any idea about how well the mapping units actually describe your sites? And could the scaling, the difference in scale have changed your results at all?
Yeah, that's a good question. We had pretty low resolution to the mapping units. I understand that in the US they tend to be on the order of a couple 100 meters, and ours were much larger, so it had pretty low resolution. But so they didn't match quite as well.
I had a question about the density of infected reservoirs, I guess. So there's this alternative hypothesis, at least for West Nile in the western U.S., that drought decreases the bird population, which increases the density of infected birds, and then increases the odds that when a mosquito bites a bird, that then there's a transfer. So I guess my question is, like, the hypothesis that you laid out was kind of the opposite of that, it seems. So do you think one of those is wrong, or do you think that it depends on the disease, or it depends on the ecological dynamics?
Probably depends on disease system. I think that the feeding behavior is quite specialized for mosquitoes and for fleas.
But I guess in terms of density, you said that your hypothesis was that the population increased when it was rainy, and therefore the density of infected individuals increased. And I guess that's my question about the relationship between the total population and the density of infected.
So I think the density increased with the soil parameters, but the mean annual precipitation has more to do with flea survival as opposed to rodent population, which I think is governed by soil parameters.
So, based on your research, do you believe that all the poaching in Kenya has insignificant or significant effect on infected rodents?
Well, I think that, we didn't find that, experimentally the removal of large wildlife leads to an increase in rodent abundance. And so if there's selective poaching on wildlife areas, I would think that would have a significant increase in the rodent population for those land use types. So if poaching is selective towards wildlife areas, that might be like kind of a double whammy in terms of effect on the rodent population.
Whitney, thank you for your incredible clarity of presentation. It was just wonderful to listen. And I just had a more conceptual question. Where a lot of this work is looking at plan change as the driver for disease. And I'm just kind of curious more from a socio-ecological systems perspective. In either of the regions where you work, do you or others see examples of how human behavior on the land might be also part of a sort of systems control on disease, where it's not just land change as a unidirectional driver, but actually human behavior on the land maybe includes people moving away from areas where they're seeing examples, sort of local knowledge of how humans are responding outside of medical intervention to change disease patterns in kind of a regional context. So I don't know if that's something you think about or something you've seen examples of or think about.
Sure, yeah, that's a great question. So in terms of human behavior, that there's, especially by land use, there's different behaviors by land use that might predispose people to become infected, like with rodents, for example. So you might have rodent control practices that are prevalent in some land use types and not others. Or people might be moving away from areas because of rodent problems. We know that rodents are major agricultural pests, so there's a lot of behavior of rodent control around farming areas and such.
I have a question about the anthrax vaccine.
Mm-hmm.
And does that confer protection for just 1 year? Do you have to re-administer it each year to the same animals?
You have to re-administer it. Actually, the protection is only conferred for about 6 to 9 months. So really timing that really well with the monsoon would be ideal. That's it. Thank you, everyone.